everyone's here. Yeah.
Okay.
Yeah, I think just wait. You'll just start the recording or wait. Yeah. Okay. So hi everyone, welcome to Tuesday's reading group. Today I'm going to continue introducing AlphaFold. In the last quarter, it's actually spring 2021. Last quarter we mentioned the AlphaFold 1, and we actually have some guests on AlphaFold 2 and how they are working on the proton folding problem. In this year, August 15, Deepmind's paper AlphaFold version 2 is out and published on Nature. And that time, we have a chance to see how they are solving the problem, proton folding problem, which is a decade-long challenge in biology. So for today, since it has been a long time, we last introduced this problem, I'll spend a couple of minutes to do a quick recap on the proton folding as well as the AlphaFold version 1. And today we're going to mostly introducing the important details on AlphaFold 2. If you ask what is the relationship between AlphaFold 1 and AlphaFold 2, I'll say they're almost totally different. And except the part for feature engineering, where they are collecting information from protons and protons amino acid sequences. Once into the modeling, it seems change a lot from the previous version, and almost nothing in common, I'll honestly say. But let's get started with today's agenda. So I'll use a couple of minutes to introduce again about the proton structure prediction problem and AlphaFold 1, which is a model based on CNN back to 2018. And most of the time we'll point out to AlphaFold version 2, where they are using an evoformer or a transformer and attention-based model for proton prediction with I would say, very complicated model for proton folding. And afterwards, I'll point out some additional and external resources. Interestingly enough, on the exact same date, July 15, 2021, another paper from Science, researchers from University of Washington, Seattle, and also other institutions are publishing another work. I think it's called, I forgot the name, but I think it's another model for protein architecture prediction. Oh, it's called Rosetta Fold, with a certain design structure on that model. So I'll also point out some external resources and the blogs and media posts I have read for this reading group. And I also collected in a Google Doc I'll share later after the reading group. So for today's paper, we are talking about the second item. The paper's title is Highly Accurate Protein Structure Prediction, published a month ago. And two other related paper. The first one we have introduced in the last reading group, and third one is the publication on Science I mentioned just now. So I'll point out to protons. So last time we mentioned about the importance of analyzing protein structures, and the situation is the building block, the building blocks of a protein is very. I will not call it simple because I actually don't understand a lot of it. Maybe we can point out if or adding more information on that as last time. So for building up a protein as a chain, it only has like 20 amino acids type in a string, but they are in one protein there are typically hundreds of amino acids, which is from 20 types of them. And for number of individual or different proteins in human body and the entire Earth, there's a lot, and not every one of them or most of them we cannot afford to know the structure by experimental method, which is really time consuming and highly cost. So we might be interested in certain proteins that are of great importance. For example, during the pandemic, the viral protein or the translated protein in the human body, once it enters the human body, has been fastly identified about their sequencing and structures. So, but not every protein can have this same treatment, or our human beings cannot afford to do it. So, but protein functionality and folding is important, because there's some research on that saying the protein, although the sequencing is relatively easier to get, but the most important information hidden in one protein is actually in their structures. So that's why a great research effort has been put since a decade ago to find an efficient way to predict how the protein folds and how it can potentially find drug co-host or some other applications in medical domain. This is a picture of how in experiment a protein can be measured in 3D structure. I believe the general idea is to like put a sample into a very low temperature and make it relatively easy to observe the atom position and put it into the structure information. Okay, so in one sentence, it's very important. Not for a near future or for a long future. And now we have a quick recap on how the protein are structured. So there are different levels. The first level is primary features, is just the sequence together with the amino acid type. And once the amino acids are combined into a peptide, and they are combined or connected by the peptide bond. However, the rest of them, in that case, we can call it a residual of the protein. It's actually different and will be connected. And you can see something similar to this. And it is a sequence with some 3-letter acronym to represent each component as the protein type. So this is and also scientists are naming each protein within the alphabet, because there are only 20 of them, to naming the type of the protein, of the amino acids. So there are 20 of them, less than 26 in English letters. So in that case, we have pretty much a sequence information of how protein is constructed. This is called primary because this is the most basic information of a protein. In secondary, it's some standard or commonly observed helices and sheets inside of protein structure. It's more like a substructure, but not fully disclosed of their 3D structures. It is like a feature for very typical substructures. So it's not the complete folding structure, but some typical structures that can be observed in multiple proteins. They're either in some helices or sheets. So we'll call it alpha or beta structures. And the tertiary structure, the level 3 structure, is what we are trying to predict. So for which typically to predict the spatial location of an atom, and more importantly, the peptide or the backbone structure. So there are the residual structure with other atoms in the side. Sometimes it might not be as important as the backbone position. So in the later AlphaFold 2 structure prediction module, it can predict both the backbone position and also the side or the residual position, which is you can infer, or you can conclude that it is a model that can predict every atom position in the compound. The quaternary structure is a combination of a multiple peptide, and a single polypeptide is more like a combination of them. In short, the task of it is to predict from the sequence to a 3D structure, or in short, from the level 1 feature to level 3 feature. And in terms of the backbone geometry, we also have one slide to talk about in terms of structural prediction. So in the AlphaFold 2, the general there is it's called backbone frame. In that backbone frame, it determines a dimension of residual, it's the dimension of number of residuals and a rotation and translation parameter. So that is the way that we predict the backbone frame location. So that's again, it's like when you do the to parameterize the rotation. Typically in this work, they are using like a rotation parameter, which is a 3x3 matrix, and also translation is a 1x3 parameter to describe how one residual is located related to in the location of another residual. So, and also in terms of location, later in AlphaFold 2, it has a definition of local frame, which is defined locally in the local carbon atom, or globally, which is the location from more like an absolute location from the very first, the beginning of the sequence. So you can have a local and local frame or coordinate to determine the location of that. So that's a little bit about background. Any questions or? Just a little bit information about the entire challenge. So the challenge is called CASP. It is held every 2 years and also has a special session this last year in 2020 for COVID-19 track. And in terms of evaluation, there are multiple measurements to tell how the prediction is good or not, or whether the prediction is good or not. Generally, you can imagine that's a commonly used evaluation metric is called global distance test. Which roughly it's a L2 norm distance of different atom location, or the backbone location between the difference between the ground truth and your predictions. And another way, which is called the TM score, is put that the Euclidean distance into the a certain rank of numbers. So make it between make the rank between from 0 to 1. And it's more like a similarity score. 0 probably nothing to 0, but 1 is exactly same, and 0 is nothing likely the same. So that's some evaluation about to predict two protein structures about their atom location. In terms of, this is related to how we know the proteins. As I mentioned last time, most of the protein we can only know the sequences, and there are hundreds and millions protein around the world that would probably only know the sequence information. A small part of them, about, I'm not sure whether the number will be updated right now, but around not less than 1 million will be in the structural database, which is called PDB. And in that case, there are some information and complete information that have been detected or can be experimentally verified for their protein structures. And in the AlphaFold 1 and 2, those variants are using sequence databases and structural database as well. And you can consider a structural database has the ground truth geometry or the 3D location information. So that is we can consider that's a supervised, fully known, well-known database, and the others are we can only know the sequences. But there are some training strategy in AlphaFold 2. It's called self-distillation that can involves in certain to treat several highly confident predictions as their ground truth data, used as a ground truth data. So that's the part I will mention that later about training strategy.
What does structural database entries look like? Is it specifying 3D coordinates, or what do they say regarding the structural database information?
They have the information of their 3D coordinates. So
3D coordinate, XYZ coordinate.
Yeah.
Okay.
So the entire full. So I have a couple of slides about the AlphaFold 2. After the paper was released, they also released their the data set. I think they collect a large base of data set and also release that, and as well as code base. The data set itself is about, I think it's about a terabyte large in terms of their size, the sizes, and not everyone else in the around the world might be easily to replicate your results. So I will point out the resource slides later, and we can see how. And also they also provide their prediction for a lot of proteins. So we can, at the end of this this reading group, we'll show some resource slides talking about how they are gathering their resources from PDB and other sequential database. Okay, let's get back to AlphaFold 1, but before that, let's just give you a sense of how AlphaFold 2 are advanced compared with AlphaFold. Is a lot, and yeah, and so I see from some forums saying that talking about, so what is the like a threshold for reliable and usable, or giving the sense of how the scores look like. So I probably don't have the, let me check. Yeah, so you can see the global distance test as about, if the number are about over 80, so we can generally infer most of the positions are correct, and if it is over 60, then the backbone position is roughly right. So that's a impression of a threshold 60 and 80. So in terms of AlphaFold 2, it's over 80 and get a really astonishing performance.
What's the difference between backbone position and the global structural position? Exactly what additional information do we need in order to get the global position from the backbone position?
So in terms of the prediction, there are AlphaFold 2 is capable of predicting every atom, not the backbone atoms, but also the side, the residual part. So the backbone, so there are several evaluations that primarily focusing on whether your backbone is correct, because the backbone itself construct the primary, the most important features for a protein. So for different levels, AlphaFold 2 can do every atom position prediction, but some of them are only trying to predict the backbone, which is the carbon, nitrogen, and hydrogen and oxygen atom in the peptide bond. So
could you remind me what a residual was?
Sorry, what's that?
What is a residual exactly?
Residual is more like after the amino acids combining with each other. Can you see the R group here?
R.
So yeah, the R part will be the residual of the amino acid, which is the part which can be which is the indicator of this type of amino acids combined.
I don't see any R. Where is R? Could you point where R is?
Yeah, let me find a way that with a pointer. Is there a pointer here?
It's in the big, I mean, round circle that's in the green.
Yeah.
Oh, that one.
Yeah. So the R is, sorry, the R is different here for different type of amino acids. There are a total of 20 of them. So this part is the, yeah, I hope this is based on my limited biology information. Some prediction on the backbone will be only to predict this the position of the peptide bound, but some more advanced tasks would be predict every atom position, including the residual part. So for different residual, the R is different in the molecule.
I'm still not sure about, because I know nothing about this. Amino acid includes R or it doesn't include R?
It does.
It includes R.
It does include R. So when I say A or A amino acid or ARG amino acid, it includes a particular R.
Yes.
Yes.
Yeah, there's 20 different type of the R. And then that's why we say there's 20 different type of amino acid. On the backbone, which is the hydrogen, the carbon, and then that part, basically, you can think of that they form a rigid, well, more like rigid chain, but that chain forms 3D shape. That's where the shape comes from, the global shape comes from. But so if you look at a specific amino acid, right, each one have a side chain, right? Like you can see connected to the backbone, right? And then this side chain has 20 different types. That's where your 20 different amino acid type come from. And then because of difference in the shape, the side chain, they may have a different like chemical, physical property. And maybe some of them like water, some of them really, repel water, and some of them maybe positive, slightly positive charge, and some of them slightly negative charge, and some of them maybe more like acidic or some, so there's different properties there. And all because of the side chain difference, which is our part. But it's more difficult to predict the location because you can think of even the backbone doesn't move, right? The R, you can twist a different angle, right? So that there's some more flexibility there, and it's harder to predict.
Okay, just one more. Here, when I look at the amino acid, I see C and H. Exactly how is that amino acid connected to that like CO and H backbone connection?
I think I want to, I don't have a size, but I feel like this one is, it could be helpful for a case that the gray one is the backbone that are the carbon compounds and nitrogen compounds that they are connected, and their residual part are the orange part. So this is my guess. So I'm not sure which amino acid it is, but this is the gray one is the backbone, but their residual the residual part will be on the side.
I see. What is the blue and what is the red?
I think
the blue
red
probably
either nitrogen or oxygen.
Yeah,
I think it's nitrogen.
Oh,
yeah, and gray is a carbon atom.
Yeah,
and
different type of item.
So the side orange thing is almost like all these amino acids are almost like side branch.
Yeah, it's a side branch. You can see the side branch has more degree of freedom there.
And this yellow, the orange thing, the first like atom, exactly what is that one? Is that always a particular atom in the amino acid, or it depends on the particular amino acid?
The first orange.
Yeah, the orange that is connected to
the orange part could be. Yeah. So if you look at the blue, the gray, the red part, right? This backbone. They for all the amino acids, they are the same. They are all with the same structure, right? The only difference is this side chain, right? So for different amino acids, there are different items, right? And there's different geometry, right? Structure, and because that would give them different property.
Could you, Junheng, could you go back to the amino acid slide, like slide number 14?
Yeah.
So here then C is part of that gray, or C is not part of the gray, the central
C is part of the gray.
Part of the gray. Okay. I think I got it now. Okay, good. Thank you.
Okay. Thanks, Wei, for helping a lot. Yeah. And a quick reminder of the first AlphaFold. If you can still remember what we have discussed last time. So the AlphaFold version 1 is a convolutional neural network. So in terms of how they're going to prediction, this is the model overview. So you can imagine this. The input is something like a picture, or pictures has a width and height with the RGB channels, right? So here it's the length. It's a square matrix, a square tensor, not square tensor. L by L is the length of the protein sequence, and with a certain channels of the feature. The features are constructed from a wide range of resources and something we have known about the structure. So that's more towards the bio side. But in terms of the input, it is L by L by dimension by channels. The channels are the number of features under this kind of under this. You can imagine that's a protein image, and the input is L by L, which is now the number of protein sequences. And there is a convolutional neural network, just like in the image domain. And to do this, transformation from the original feature input to a distance and distortion predictions. So the output would be exactly the information needed for distance and torsion information. So there are some other structural models that based on the distance prediction and torsion angle prediction to finalize their final prediction of structures. But this is a general model structure of AlphaFold version 1, which we might skip a little bit for to save the time for AlphaFold 2. But generally, the pipeline of it is this is a original paper for Nature, and Nature will just have a high-level guideline here. The yellow part on the left is the feature engineering part, and inside of the green box, you can enlarge it to figure B. That's the part that with a typical convolutional neural network structure, and then predict the following output: torsion distribution, distance distributions, and they're using that information to predict the angle and doing the back propagation, and also determine a loss function about this about the distance between the ground truths and our predictions. So this is a general pipeline of models for AlphaFold version 1 using.
I want to ask some more questions.
Sure.
Here I see 4 red boxes: torsion distribution and distance distribution, van der Waals, and potential. So let me start with the distance distribution, because I think that I understand that best. So the distance is, so here these distributions are for a training data set. We are already given this distance distribution, right? We can compute it.
Yes.
So in some sense, we are training against the distance distribution that we already have for the training data set. Okay, and then here the distance distribution is distance between what? Distance between individual amino acids?
Distance between, my understanding, probably not very exactly correct. I have to be honest here, that is a little bit out of the biology geometry here, or chemicology here. But my understanding of their torsion and my understanding of the distance distribution is the distance to another residual or another amino acids in the sequence. So if you can see this part, this is the sequence from 1 to L, and the other dimension from 1 to L, and this is a distance matrix. So they want to output a distance matrix pairwise.
Right. So here L is the number of amino acids, right?
Yes.
L is number of amino. So we have this L by L distance that indicates what's the distance of the first amino acid to the first one, second one, third one, etc.
Yeah.
Okay, so that is distance. What is torsion? I forgot what the definition of torsion was.
My understanding of torsion is something related to the angle, but I'm not sure in terms of their implementation is I think the figure shows the distance distribution, but I'm not sure how it could be different for torsion prediction. But my understanding is it's something related to the angle prediction as well, in terms of between different amino acids, or they're in other coordinates or geometric location wise. So I'm not fully sure about that. So that's part I probably cannot answer. I'll be honest on that.
So torsion, Yeah, maybe Wei can answer.
So for that matrix, right? So they build it at the atom level, right?
Right. Oh, sorry. This one
Not-
is on the residual level.
But then when you talk about the distance, which I mean, which location do you use as to represent location of this amino acid?
I think it's I think I checked a long time ago. I think I checked. They're using there are two carbon atom, but one of the carbon atom as like a red flag, yeah, alpha or some certain carbon as their representation of real absolute location of that.
In that regard, they can only predict the backbone position.
Yeah, I think so.
Because there's no information about the side chain position.
That's what I infer from the structure, but I believe there are some other other way to do, or in the AlphaFold 1 they do primarily on backbone prediction. This part I probably need to check back. Yeah.
I think the distortion is on the angle of the backbone only, because they're only concerned about the backbone.
Mm-hmm.
And what about the van der Waals force? Is it
this part I didn't check. Sorry about that.
And what about potential? What is potential here?
I think that's the prediction. The final prediction for given the distance distribution, torsion distribution, they have a finalized version for the output prediction.
So the potential is the final overall potential of the molecule.
I think so. Yeah.
Why do you have phi and psi there?
I think that's the two major torsion angle of backbone in the peptide bound.
Did we define what phi and psi were before, or we didn't define it?
I feel I think something related
somehow
to
related to the angle.
To this angle, but for me I'm not quite sure about yeah. So
the potential is related to the energy, right? Because when a structure, if you think about the structure need to be stable, which means its energy need to be minimized in some way. So that the structure can become stable. The van der Waals force is basically just so you think about the force between the atoms or molecules, right? They're either repel each other or like attract each other. Those are the force. So, but I don't know how they represent the force here. And
here the potential, is it given as more of like a function for the training set, or is it just a number that they know that in a stable state that is the overall energy level?
I think that's just a prediction on I need to check that. Sorry for not including that detail. I think it's some parameters or some prediction functions there to have a final saying on given the prediction out of torsion distance distributions. It has been a long time. I remember the detail of this. Yeah. So.
And lastly, regarding the blue one, initialization, I think is parameter initialization. Gradient descent is taking gradient descent to optimize. What is structure pool?
So that is the part I feel like is for me, I'm not quite sure about that, because in Deepmind they are having a certain biologist or biochemist are working on some final restriction or some chemical structure violation checking rules. So I believe that's related to some post prediction after making the prediction in the machine learning part.
I'm guessing,
so
I haven't read the paper, but I'm guessing the structure pool is a way to confine the search space. It's just because we already know maybe around the 200,000 protein structures by this crystallization. And then that's give you somewhat an idea about what the distribution of protein structure may look like. And then that's, if you want to predict or determine the structure of a new protein, right? And then if whatever, based on whatever feature is look like similar to an existing protein, right? So the model should learn that and then use that as a guide to determine this protein.
So that makes
the structure of the new protein. That's my guess, because I didn't read this paper, so I do not know what
right,
they
so that makes
really
sense. So in some sense, in the machine learning terminology, sounds like we are adding regularization to have some induction bias.
yeah, something like that.
Yeah.
And that's what they, yeah.
One last thing is, why do they say adding noise?
This, I think for that part, I can point out something probably similar in AlphaFold 2. They are having a little bit auxiliary training in terms of the prediction, and I'm not sure whether that is related to adding some for some prediction. They are adding certain perturbation on the prediction to refine the model. I'm not sure whether this adding noise are referring to that process. But I can mention that in AlphaFold version 2 some strategy related to auxiliary loss.
Okay, thank you. I think I got it now.
Okay.
And I have to introduce this again, although Wei knows more than this. This part is related to feature construction, because that's something we need to know for AlphaFold 2 about a mutual enhancement or a iterative training towards multiple sequence alignment, or the MSA part, to the pairwise modeling part. Let me point out the general idea of sequence alignment. So like in AlphaFold 1, when we are doing the sequence prediction for a protein, we're not just using the sequence information alone. Actually, we are doing a little bit, knowing we have a protein database, and we can do a search on by certain criteria on searching similar protein sequences can be occurred in another species. So in this case, in this MSA module, they are doing it's part of still in the input stage. They're trying to go to search for similar sequences and return some of the top similar sequences that are might be close or with a large overlap of similar segment in the protein sequence. So it's more like a searching for similar sequences out of the known protein base. So in that being said, we are not just having a new sequence just doing the inference. At the same time, we are actually searching for some possibly some known protein sequences, some known templates of their 3D structures, and utilizing that information as well. And MSA part is a very important building block in terms of the input in AlphaFold 2, which I will introduce very, very soon. So short summary of that. This is more like return a series of similar protein sequences with known information. Let me jump to AlphaFold 2, and now let's get some general idea. The glance view of the entire model architecture. So let's go to the input embedding, encoding, and the next will come to when it comes to final prediction. So the first part will be, I'm going with the pointer. So there are two parts of input. One is called MSA matrix or tensor. The other side is called pairwise matrix. So what is in the pairwise? The pairwise is a matrix with the dimension of number of residual and number of residual. So it's a square matrix. Well, theoretically, it's a tensor because each entry in the matrix is actually a channel length features. So it's another dimension there in the back. So, but generally it's a residual by residual. So it models the interaction between the residual I and J. So it's more like a confluent matrix. The MSA module has the dimension of number of similar sequences and number of residuals. So the residual will be chopped into the same length to have a hyperparameter for the chopping or some feature engineering on that part. And the sequences is the number of similar sequences we have found in the protein database. So in this case, I'll name it the MSA dimension is S by R. S is the number of sequences, R is the number of residuals, and the dimension of this pairwise matrix is R by R. And now we have this ready, and it will be ready into the encoding part. There's a huge effort of constructing the MSC and pairwise matrix, gathering multiple sources of information. There's 62 pages of technical report for this paper, and almost 10 of them are defining what are the features or indexes they are using. But for now, we can just think about the input as S by R in MSA and R by R in pairwise matrix. So that's the input part, and in the title, it has been named under embedding. So which means there is already a feature transformation from their original values to a certain to the, I'll say, the main encoder module. Before that, the embedding side, we already have the M by MSA, which S by R and R by R as input, two side of input, and we use those two sides for mutual update in the main chunk encoding part.
What are the channels like for after the pairing?
What are the channels like? So let me point out to, let me first get to this part. So in their technical report, they have this slides for the entire feature engineering to the pair representation and MSA representation. They have some additional extra MSA representation and something like this. So that you can see the dimension is actually a tensor R by R, S by R, and the CM, the CZ are dimension of features of channel. Sorry, number of feature channels. And what are the features? This is out of the machine learning part, but I can point out to, oh, sorry, let me. It should be in this, yes. So in this 62 pages, they have the data pipeline here and featureization and model inputs. Let me point out some important features here. Sorry about the, yeah, here. As I mentioned, there are some feature transformation already from the raw features. So here, some features, important features, one-hop representation of input amino acid sequence. And there are also their dimension. Those are, the dimension might not match the final version of the channel, but they are constructing certain way to a lot of features concatenating with each other. Not just concatenating, but also doing transformation. And they are, this is the complete table, but important features, I believe they are here. The residual feature and the MSA feature, something like this. That's out of my domain knowledge about how the features are. But I think some straightforward features like given the features, what are the amino acid type, right? Those are one hot representation of one of them is that.
But this is for pairwise information. No?
Yes, I think so. But
then
there are
what
some
amino acid are we talking about? Is it like 20 by 20, so 400, for one hot encoding of 400?
I think that's, if you are saying pairwise channels, I believe they are concatenating the pair of amino acid in kind of concatenating the two of them. I think that's one of the feature engineering in AlphaFold 1, but I'm not sure about in AlphaFold 2. But I think they are stacking features together from both amino acids, if that's the pair in the pairwise sense. In MSA, it's a sequence, it's a number of species by number of residuals. So that's I believe there are a lot of engineering work in terms of in the entire feature model. There are a lot. So as you can see there, yeah. So that probably I cannot fully understand all of them, but I do consider there are a huge amount of feature engineering team in Deepmind just to focus on how to make it more salient and expressive. Shall we continue?
Yes, please. Thank you.
Okay. So in terms of now we comes to after we got input, you can S by R by R, two side, and we comes to the sequence residual edges, which is the part that two part of information are interacting with each other. So the logic is here like, so in the residual part, we try to analyze the pairwise interaction between residual I and J. And we can also have the information of how pairwise information of pairwise residual I and J can be interacting with each other by the information from MSA matrix. So which means if we, or another sense of understanding is that if we notice the residual I and J, like this red one and orange one, I and J has some feature interaction observed from multiple sequences, the MSA part. We might input that information to the residual matrix, which is, for example, the red to orange, which is this entry. The information might need to be updated from the 2 rows in the MSA part. So, and also the updated residual information can also be reflected or being forwarded to MSA again. So that's where you can see update pairs and update sequences. So it's more like an iterative process. They are mutually influence each other. So this is the general logic, like the MSA and the residual pairwise information are the 2 salient information that are really beneficial to the final prediction of the structure. And this trunk part would have those attention-based model to update those things iteratively. And now we are coming to how they are updating iteratively. But this is the general sense of what we are trying to do here. So this is a more clear version of how they are doing that. This is the entire AlphaFold 2 version. So the evoformer or evoformer here is the part to do some encoding. From the input and output are exactly the same. The MSA and after the evoformer blocks will be the updated MSA. And pair representation after the evoformer, it will be an also updated pairwise representation. But in the evoformer, the information from both sides are actually interacting with each other. And once it comes to structural model, we will have one single row, which is our target prediction. You might see this is like we want to predict the protein observed in the human species. So now we try to predict this more like in the inference stage. We given one single representation R by C, which is the residual by channel. and together with their RRC, which is a pair representation, into the structure module.
Can one of you reach out to Junheng?
Okay. Let me directly go to the part. It might just another computer just accidentally corrupted. Okay, let me go to the slides we are having here. Okay, got it. Yeah, so this is the part that we are, the evoformer and structure model are the main machine learning part for an attention-based, or say using attention-based part to attention-based model to jointly modeling both sides. And once they come to structure model, the input is, as I mentioned, the input is the single representation with the pairwise representation matrix, and output the rotation and translation of the backbone, as well as the position of other side or residuals we have. So the 3D structure will be actually outputted, generated during the structure model. Let's see, we have enough time to go over all of the two, but let me just, let's primarily go, first go to the evoformer part. Yeah, this is the idea of evolution how they are defining evolution transformer. Just like the key idea here is to design a transformer. The first transformer is applied on MSA, and the general assumption here is after you attentively learning something from the MSA transformer, two rows of information can be later integrated into the pairwise representation. And then pairwise transformer is useful as well, and they might getting that information back to pair of residuals to MSA representation to update their information in that MSA. So this is the general pipeline to how they are iteratively learning those two. This is the decomposition of how they are in one of the evoformer block, how they are do this entire encoding process. So let me just point out, let's go to the input and output. Exactly the same for MSA representation matrix and pair representation matrix, with the dimension of SRC, RRC. R is the residual, S is the number of sequences, C is the channel. Note that those two channels are different in different dimensions, I believe. And in terms of every block of the evoformer, they have 8 sub-blocks. So the important module we are going to introduce today is called row-wise gated self-attention with pair biases. Because the row of sorry, I think that's the column of the two would be regarding to a residual I and a residual J. And with that part being said, we want the pairwise information of residual I and J being imported from the pairwise representation as one of the mutual mutual learning motivation we have discussed. And for column-wise self-gated attention, then we don't need the pairwise information because that's relatively on the sequence level in the dimension of sequences. And then they are doing some certain transformation and input the information back to pair representation. And in the pair representation with the input information from updated MSA, and now we are input with 4 blocks of triangle-based update, triangle update. It's called triangle update using outgoing edges, triangle update using incoming edges. Those two are relatively similar. We will see the edges. What are the edges here? And another two called triangle self-attention around starting node, triangle self-attention around ending node. So those two are another pair. So those those two are similar. Let me go back to this laser again. Those two are relatively similar, and those two are relatively similar. And the final transition, and after a final transformation, we get the updated pair representation, and as well, a while ago, we also have the updated MSA representation.
I have no idea what outgoing edges and incoming edges and what is triangle update.
Yeah, I will explain that in later slides.
Okay.
Yeah, what are defined edges? The short answer is that it defines as a graph here. I and J is as a complete graph between the residual I and the residual J. So there's between residual I and residual J, there's a two edges between those residuals, and it's a complete graph. And we try to use that kind of residual graph, which is a complete graph between residuals. But I will go to that part, what we call a triangle update. And what are the edges here? Yeah. This is our algorithm block for the exact same process, where they have a lot of block here, another one transformation adding to another. So let's go to the MSA part. Well, it's a little bit complicated in terms of for every row, for every block of this module, but we will decompose it to a couple of them, a couple of representative modules here. So AlphaFold 2, the first component is row-wise gated attention. I would refer to it as more like a I will say like it's a little bit like a cross attention from both sides, but the general computation flow is like this. So the input is one row of gated attention, but with the one single row, we have the dimension of R residuals and with channel Cn with the number of channels. And there are the rest of them would be attention method here, but there is a certain part which I'll point out separately here, as pairwise presentation will be inputted as a certain kind of pair bias into one of in the middle stack. So let's see the model from the left to the right. So the right will be exactly same in terms of output, exactly same dimension with the R by CM embedding. So which is we are updating the rows here. For one single row, we have the attention part. So basically those are by the way, those RQ and RV and also this queries, keys, and values, those are actually all the R are actually the same dimension as R. There's no different there's not different number here. In their implementation, they're all RQ, RV, and they are all the same. So all into the same value as R. In terms of the encoding part, this is a typical attention design for that. So queries with a dimension of R number of head in the attention are HC, and key dimension from their linear node bias, I think linear node bias model as a R, H, and C, which comes to a dot product affinities as R, R, H with their out product computed by C channels. And similar going to the value part, and together with the pair bias added to the dot product affinities, we come to a softmax layer and comes to final tension weights with the final output of RHC. And then finally getting from the input again, another thread of computation graph with the linear and sigmoid, linear node bias and sigmoid layer coming to a gating RHC, and also will be multiplied together, and final layer from the head and C here to the final CM channel. So that's the entire process for treating the row-wise gated tension.
I need a lot more detail. Let's start with a dot product affinities, the red one.
Yes.
So
here,
that
right?
one is that one dimension is R by R or whatever, L by L, right?
R by H. So there's n hat in terms of the
Right, R by H type. What? Yeah, L by H by C, number of channels.
No, there's no number of channels.
Oh,
There's
there is
a
no
dot
number of
product.
channels?
The dot product here.
Oh, these are
They're
all dot producted. For each head,
Yes.
these are all dot product. Okay, so that is fine.
Correct.
So now we have that, and then we add the pair bias to it, and then we take the softmax. Like when you take, what does softmax mean? Row-wise softmax or column-wise softmax or entire matrix softmax?
I think that's the entire matrix softmax.
The entire matrix.
Yes. I think let me.
It says RV, so it is something.
Oh, I think probably let me see. This part I suppose it might be it might be column-wise softmax in terms of their I probably need to check this to give a final answer, but I believe it's a column-wise softmax here.
So the column sum is 1 then after the output.
Yeah, I think so.
Yeah, column sum is 1. Okay, so fine. And then now we have this matrix, attention weights matrix. Or stochastic, it's a stochastic matrix. Column sum is 1. So now we have that. And then you are saying that now to that stochastic matrix, we are multiplying the values. And then we get the output. I see. So what does it mean then? Because
I think that's a good question. Why is designed that way? Or
right side, left side.
So so the general cases here, I personally believe in terms of the attention, they are designing in a row-wise and column-wise, and the column-wise will be exactly similar structure without pair biases. That's the two part of the MSA. Yeah. So in terms of the and also they have a separate gating part using this exactly same input R by CM into the most upwards here, upmost gate using like a structure, call it a gating, and then to multiply by the output from the attention weights.
Right,
so that's the part.
Element-wise multiplication.
Yes.
Between
this
gating,
one is element-wise multiplication.
between gating vector and the output vector.
Correct.
That's and also we pass it through another linear layer and we get the final value.
Yes, back to the original dimension R by CM.
Right.
Yeah,
Okay. I don't know what is going on, but at least I know what is being done. I don't know why, but something is being done.
Yeah, so in terms of why, you might have this question a lot. Like for the general MSA to pairwise mutual enhancement, I can feel their justification or their motivation is to doing that. They have assumption that pairwise information can be learned from MSA, and it also have a reverse effect from the pairwise information to the MSA. So in terms of why they are designing a row-wise gated attention, so it could be they probably have tried a lot of other cases or simpler models, or even more complex models. But I feel like this is the way in terms of row-wise gated attention with the pair biases into that. So this is, but I think something will be transferable from this proton folding setting is we have two information resources, two information sources that want to have a fusion in terms of modeling, and with a certain bias from the other source. This is potentially a modeling like how we can construct the encoding or transformation in terms of their with the intuition of how they can be influenced with each other. In terms of column-wise gated attention, it's exactly the same structure, but without pairwise information. So you can imagine there's no added thing here, and it's just the upper side without the blue boxes, the blue arrays here as pairwise attention. The next step is MSA stack to pair stack. This part is relatively easier to understand, that we try to have the two column information from I and J, residual I and residual J, and put into the model and assign it to a CZ, which is the channel number in the pairwise representation. In terms of in terms of the process, that generally having the two let's say two S by CM vectors and into the linear outer product and mean layers into the final linear layers from the C by C to final CZ, which is the number of channels in pairwise representation. So that is where the information are transferred from MSA to pair. This is the encoding process. I would say that by an outer product mean. I see that we have about 10 minutes left. I'm not sure we can finish all the pair transformer thing, but let me give a general first sense of where's the graph, where are the edges and graph here. So this is the pairwise representation R by C are actually constructing as a complete graph between every two residuals in the sequence. So that's a complete graph with the, this is example or demo in the left side with the residual I, J, K as their complete graph, right? They have the edges from I, J, J, I, and it is considered in a direct graph. So I, J, and J, I will be different. So that's where we will have outgoing and incoming starting node and ending node. The definition is under a direct graph setting. So the graph itself is the complete graph between every two residuals. Since the general length of the protons, one proton, a general length of that is typically hundreds of them. So the graph, one graph for one particular proton is not large that much, because the number of nodes here, the number of nodes as residuals is about hundreds of them. So that's for the corresponding edges in a graph and how we are defining a graph. And then the attention is something they're calling triangular attention, applied iteratively for 4 settings. And the upper side are called multiplicative update for outgoing edges and incoming edges. The lower side called a self-attention around starting node and ending node. This could be a little bit difficult to understand at one time, but let me just finish the first thing, the outgoing edges, which are when we are updating the edges under the entry of ij, we are considering all the outgoing edges to other residuals at the same time, which is we are considering the influence of the impact of ik and jk, which are outgoing edges from the node I and j. In other sense, we are trying to update ij with ik and jk of all and also all other residuals. So the outgoing are towards to the ij entry, and incoming are just the reverse direction. And in terms of, we're going back to a starting node and ending node whenever we are going to analyze how we are going to introduce how the model are going to compute. So in terms of triangular multiplicative update, the input here in terms of analyzing outgoing edges would be two rows, row I and row j, and all the vectors or all the elements in those two rows as outgoing edges for ij. So the two rows are the outgoing edges entries for the ij. And update process, this is a computational flow with called left edges, which are the edges on outgoing edges from node I, and the left edges are the outgoing edges from j. And then going with this computation process, with the linear, sigmoid, sum, the layer norm, also another linear, and yeah, so basically they are working with a and also with the specific calculation, and finally updated to a CZ vector on this. So there you can see the output will be a CZ vector. Z is the number of channel in each entry of pairwise one entry of I and j. So you can see the inception field or the input field would be those two rows. So that's why they are calling triangular multiplicate update. In terms of the incoming edges, will be exactly the I and j column and doing the same thing. So that's the part. Those two updates are actually relatively similar with the same architecture or computation in this figure. In terms of the intuition behind it, I think my understanding of this is they are specifying several motifs in terms of the graph, and they are updating on the way they believe the most salient or most beneficial to the proton folding task. However, I'm not sure whether there are possibly other triangular attention can be applied into other motif, but this is the part they are presenting as the four sub-modules, and two of them right now we have already introduced of them, multiplicative update, and the other thing is called triangular self-attention, which is again more on the self-attention around the starting node ij, and the input field will be one single row, not two rows collectively, and then doing I would say you might feel familiar about this entire workflow on the attention-based computation. Well, it's similar to the MSA part, still with the general attention mechanism with the query, something with the query and edges and something like that. So it's something we are, I think it's a lot of computation in terms of customized computation based on the setting of pairwise representation. Yeah, so in terms of the calculation here, the triangular self-attention are doing with something similar to the previous row-wise attention. Or row-wise gated attention in the row-wise gated attention MSA part. And that will also be a similar structure on the ending node. And then coming back to those, yeah, as I point out here, yeah, this is one block of evoformer. Evoformer are going with 48 blocks continuously, and then the MSA representation, pair representation are believed to be sufficiently updated after the 48 blocks, and then they are going to do the structure model. Due to the time limitation, I'll just give a very quick introduction about the structural model. In this time, they are going to predict the rotation as the 3x3 matrix and the translation of the backbone, as well as other parameters are also defined in the side. So here I show a structure of in the inference stage. In the inference stage, how they are kind of adding one by one based on, so they are starting from the first amino acids and starting to extend the backbone one by one. So that's the part with the updated or recycling iteration of that to give a final output of a structure of protein structure. So in terms of structure model, they do have some specific design model called IPA model. It's not the beer, but it's IPA model, invariant point attention module to help determine the local frame with the global frame as well. So just one additional comment on what is the structure model input and output. The input will be the single representation R by C, which is or in the inference stage will be the target prediction for the protein, and the pair representation RRC learned from the previous module, and the backbone frames will be R3x3 and R3, which are the rotation and translation to be served as the input, and the output will be also predicting angles for all the side residuals and also the backbone frames at output after being processed by the IPA module with a certain design of attention. This is not the end, and there are a lot of them. So, but we are quite close to, because the structural module are relatively, there's only one IPA module here. Before we end, I also want to point out one training strategy here. Probably will be transferable to other setting. So they are doing a setting of first they train a model only on the PDB, which are all the correct structure information, and then going to predict the rest of the protein sequences and define. You can also have the confidence of the protein sequence prediction and select a portion of them, high confident subset of predictions to be incorporated into the next retraining process. So in the next training cycle, they have about 25% as PDB correct ground truth information and 75% from the high confidence subset predicted by the initial trained model. So yeah, so I think that will be, I'll temporarily stop here at right 3:30. And I hope the entire model structure does not confuse you a lot. I think the most question we have is why we have to design that in that way. And if anyone has some comment or some or suggestions, feel free to let us know. I think today we'll pause here. Should we continue next week about just a little bit less of the kind of a 10 or 15 slides about the tricks and structural model. So how about we pause right now, and we'll come back next week for the rest of them.